Unlocking the secrets of gene mapping in complex organisms is a game changer in the fight against devastating crop diseases. But here's where it gets controversial. Despite decades of genetic research, accurately predicting genes in organisms like fungal pathogens has remained frustratingly inconsistent until now. An international coalition of scientists has pioneered a groundbreaking method that significantly sharpens the precision of gene mapping, particularly in complicated organisms such as fungi. This advancement holds immense promise for improving our understanding and management of fungal diseases that threaten vital crops like wheat. Among the contributors is Rothamsted Research, a renowned UK-based institute deeply involved in this cutting-edge project. By harnessing a sophisticated bioinformatics tool, the team revisited the genome of the fungus responsible for Septoria leaf blotch, a disease notorious for causing substantial wheat yield losses across Europe. Their investigation uncovered critical flaws in earlier genetic analyses, which had produced wildly varying gene counts, ranging from 10,900 to 13,200. With only about a third of these genes consistently identified across different studies. This inconsistency has long hampered efforts to develop effective disease control strategies. To overcome these challenges, the researchers crafted an innovative tool that integrates multiple gene prediction techniques with authentic biological data derived from fungal RNA sequences. This comprehensive approach led to the identification of 13,414 high-confidence genes, offering a far more accurate and detailed genetic blueprint of the pathogen than ever before. The project, spearheaded by French scientists and involving teams from the Netherlands, Germany, the US, Australia, and the UK, compared 4 independently generated gene prediction datasets for the same Septoria strain. Dr. Jason Rudd, who led Rothamsted's role in the study, explained, although we observed many differences initially, a detailed analysis of each gene allowed us to reach a consensus on their most accurate structures. We also uncovered previously hidden genes and structural variants that had gone unnoticed. This meticulous work has culminated in the creation of a gold standard open access reference resource for Septoria, designed to accelerate global research efforts aimed at pinpointing vulnerabilities in this destructive pathogen. Moreover, the study shed new light on how the fungus organizes and expresses its genes, especially in genomic regions that control when and how genes are activated. Understanding these mechanisms is crucial because it reveals how the fungus adapts to different environmental conditions in wheat fields, potentially guiding the development of more effective disease management strategies. Dr. Rudd emphasized, accurate genome mapping is an essential milestone in combating fungal diseases, which pose an escalating threat to global food security. By refining gene prediction methods, we aim to enhance our ability to detect and counteract these pathogens before they inflict further damage on crops. Meanwhile, UK arable farmers can look forward to receiving independent, reliable data on the performance of biofungicides in crop protection, thanks to a series of pilot trials funded by the Agriculture and Horticulture Development Board, AHDB. These replicated field trials will evaluate the effectiveness of biofungicides against Septoria tridity in winter wheat over 2 cropping seasons, 2025, 2026, and 2026-2027. A consortium with a proven track record in delivering trusted, impartial research to support crop disease management has been appointed to lead this initiative. Biofungicides, a subset of biopesticides regulated as plant protection products, are commonly used in high-value horticultural markets, but have yet to be fully optimized for cereal crops. AHDB's lead crop protection scientist, Sasha White, noted, Biofungicides hold significant promise for the arable sector, but their biological nature means they require careful integration into crop protection programs. Most efficacy data comes from controlled environments, which don't always reflect the complexities of real-world field conditions. The pilot trials aim to fill this knowledge gap by testing biofungicides under field conditions, where factors like weather variability, disease progression, and interactions with other spray treatments can influence their performance. Focusing on Septoria triticale control, due to its economic impact on UK winter wheat and the availability of promising biofungicide products, the trials will assess these products on 2 wheat varieties with differing resistance levels, one moderately resistant and one moderately susceptible. This will be done within conventional fungicide programs, with varying fungicide doses to pinpoint when biofungicides begin to contribute meaningfully to disease control and yield improvements. The consortium, which includes SRUC, ADAS, and NIAP, already manages an extensive network of fungicide efficacy trials as part of AHDB's long-term research and maintains strong ties with the agrochemical industry. But here's the part most people miss. Could reliance on biofungicides inadvertently lead to complacency in fungicide use, potentially accelerating resistance development? Or might these biological agents revolutionize sustainable crop protection? What do you think? Are biofungicides the future of disease management, or just a supplementary tool? Share your thoughts and join the conversation below.